Good afternoon, everybody. I hope everyone can hear me. If you can you just use those emoticons to show me that you can hear me? We know the drill, right? So at the bottom, you have the emoji, which tells that means if you can hear me very clearly. Superb. All right. Okay. So I see that a lot of you have already joined. We are expecting few of your colleagues also to join this particular webinar. As you see, this webinar that we have, this is a complimentary webinar for all the past attendees, but we have several other people as well who have not attended the webinar before, but they wanted to be a part of this. So welcome all of you who have not attended the Eminence conferences, but you still wanted to ask a few questions from the experts. A lot of things might not make sense to you because we have covered a lot of things in the recent conference for fourth annual cleaning validation 2024. It was a very practical conference, and the idea is to ensure that you ask away your doubts, all the questions that you have. We are not picking up a particular topic of discussion, but we were getting a lot of questions on the WhatsApp group that we have created for the follow up questions. And then we thought that, okay, why not to just put you in front of the experts and you can ask the questions to them directly. All right. So before we start, means Brushali will come and introduce AV Jayakumar, Thomas Altmann, and Kiran Kota. And we have the blend of the best possible speakers that we have right now. And they will be answering all your questions with regards to cleaning validation, MACO. I saw a lot of questions revolving around the, nitrosamines during cleaning validation as well, for which Kiran can take those questions too. Um, Also, Brushali will be probably putting up few of the questions which are still unanswered from the cleaning validation group as well. So we will be taking those questions as well here. Just very important thing. Whenever you have a question, please put it in the Q&A tab. You can see the chat box that opens directly, right? Can we show the emoticon again? You can see the chat box. Below that, there is a Q&A box. Great. Okay, so in that Q&A box, put your questions there, because sometimes what happens is if you put something in the chat box and we miss it, then your question will be missed by the experts as well. So make the most of it, ask questions, but please put it in the Q&A tab. In case if your question is not comprehended well, or you want to talk to an expert as well, rather than putting the question, you can always raise hand. At the bottom, you can see the option to raise hand as well. Can you all see that? Can you show me emoticon sign? Okay, great. So you can see that raise hand option as well. So you raise the hand and let us know that if you have any, point of discussion, or you want to personally ask the question rather than typing. All right. So over to you, Vrishali, and we will be starting the day by introducing our experts. Thank you.
Good afternoon, everyone. Thank you so much, Guneet. So, before we invite all our speakers on the stage here, let me introduce you to them one by one. Mr. Jayakumar AV, he is the President Quality at Ajanta Pharma. He has completed his master's in analytical chemistry, and he is an expert in evolving sites from scratch to meet all SRA compliance requirements. He is also skilled at delivering training and conduct several audits in India and overseas. And during his career, he has dealt with all stringent regulatory authorities during inspections. He has executed several digital transformation programs, and in his current organization, making his lab and manufacturing operations paperless. Uh, Along with Mr. Jayakumar, we also have with us Dr. Kiran Kota. Dr. Kiran Kota, he is the Vice President at Harman Finochem. He has more than 24 years of experience in quality management area in uh, pharma industry, and currently is working with Harman Finochem. He is handling quality regulatory affairs department, and under his portfolio there are around 400 people who are reporting to him. He was previously associated with pharma players like Mylan, Natco, Aurobindo, MCure, uh, and such other pharma companies as well. Along with that, we have Mr. Thomas Altmann. He is Principal Global Technical Consultant at Ecolab. He has over 25 years of experience in cleaning chemistry, cleaning process, cleaning validation, and regulatory compliance. As a global technical consultant, he provides technical consultation on cleaning validation, sanitization, processes optimization of pharmaceutical, API, biotechnology, personal care in India, along with Europe, Middle East, Asia Pacific, North America, and Africa as well. In his role, he is a subject matter expert for cleaning validation, and he talks about process development, validation team, etc. He is an active member of PDA and working on cleaning validation processes updates like Technical Report 29. With this, can I invite all the three speakers on stage, please? Good afternoon, everyone.
Good afternoon.
Good afternoon.
Uh, so, Abhay, can we already take the questions which were unanswered earlier and then move on to the questions, that will keep coming up during the session as well? As you all three know, that after cleaning validation, several attendees reached out to us with these questions, and I thought I should, sort of put them up, and we can, as and when the questions come, we can start taking them. So the first question is nitrosamine risk assessment done for all products manufactured in your facility? Then there will be, will evaluation apply again?
Mhm.
Dr. Kiran, do you want to take the
Yes,
question?
yes,
Because
yes.
nitrosamine
Am I
was the
audible?
topic that you took. Yeah.
Yes, am I audible?
Yes, you are.
Yes.
Yes. The question itself is, the nitrosamines done for all the products at your facility. It's a no mandatory requirement now, as part of regulatory. But whereas the further, then there will be evaluation reply. I did not understand the statement, but however I'll just answer this according to my understanding that there is a requirement of evaluation of nitrosamine risk assessment, wherein as per the requirement, first initially we have to do the theoretical assessment. If there is any possibility of nitrosamine formation in the process, either by chemistry or by process by handling in the multi-product facilities, then we have to handle in a different way, saying that proving in an analytical way that you are well within the specification limits, and again specification limits with the results which you attain, either 10% below or 30% below or 100%. Based on that, you can apply for routine testing or skip testing or omission of the test in routine. So this is a requirement which we have to follow for all the products.
Thank you so much. Do you have anything to add to it, Thomas or Mr. Jayakumar?
I think it's very important what he just mentioned, that it's really something where you need to evaluate if you have a risk or not. So you really need to make sure and get clear of what is the outline when you are getting into a risk of forming these substances, or when there is a risk of a presence of nitrous amines, and when it's, let's say, excluded by scientific rationales. So this is the kind of information what you should get. You should not just start to make the work your way through, and then you think, oh, I do not recover solvents or whatever. So then maybe it's not really applicable to you.
Thank you so much. Uh, I hope that question is answered. If there is any follow-up, please put it in the question tab again. And Abhay, can we move on to the next question?
Mhm.
Can we use recovered solvent of a product for the cleaning of other product equipment in API manufacturing? for initial rinse, final rinse will be given by fresh solvent?
No, certainly not. The reason behind, the recovery solvent will carry a lot of impurities where your method may not be detectable, and you cannot prove with that particular, it is an additional contamination which you are making to the equipment. So that, so with this clear statement, you cannot use.
Mhm. Yeah. And I would even like to add the specification of the recovered solvent should be reviewed. So if you have a recovered solvent and you specify the density and maybe some things, but not the impurities, then you're really not getting into this subject with the right mindset. So you really need to make sure that you understand, oh yeah, I increase my risk if I use the recovered solvents. And this is why we should really look to avoid these kind of situations.
So there are two situations where you use recovered solvents. One is a recovered solvent recovered from the same product to the same piece of equipment. There are the second case where the recovered solvent from a different product using it for cleaning a different piece of equipment where another product is getting manufactured. So the risk is high where you are using recovered solvent recovered from one product to another product. The risk is very high. When you are using for the same product, the risk is minimal, but still there is a risk because, as Dr. Kiran already told, you are carrying a lot of impurities in the recovered solvents. And during the chemical reactions, you cannot predict what sort of impurities are getting transferred to the solvents, especially into the mother liquor, and then it goes for distillation. There are a lot of volatile impurities which is getting extracted into the recovered solvent. Whatever type of columns you are using for distilling your solvent. So as far as you want to minimize the risk of cross contamination from impurities of maybe now solvents are one of the critical component for nitrosamine impurities, as the studies has already been shown. So considering the criticality of that, and more molecules comes into that range of nitrosamine requirements. So as far as possible, if you can limit the use, it is better for the organization. But if you still want to use it for an economically viable manufacturing process, you should do with a lot of studies. Maybe you may have to do some GCMS or MSMS studies where you can determine what are the type of impurities present in, and that is not going to impact your next equipment cleaning process or the product processing. So that is purely based on your scientific assessment, and maybe based on a risk assessment you can scientifically justify. So preferably no, but still if you want to use it, these are the steps somebody has to follow it.
Thank you so much. Thank you so much for an elaborate answer. Abhay, can we move on to the next question? Strategy one must follow for periodic cleaning campaign validation. Can it be justified with one run? Any reference guideline for this? I think periodic cleaning was one of the questions that was coming up during the research as well. So, can we start with Jayakumarji and then move on to Thomas and Dr. Kiran as well?
So I understand from the question, this is once your cleaning validation is done. Now what should be the periodic cleaning
Period.
studies to be
Yes.
done? So it all depends upon the number of batches you manufacture, what type of product you manufacture in a facility, what is the complexity of the process involved in that. So for example, if you take an API where it is very difficult to remove from your equipment surface, something like polymers, if you are using into the process, or in a formulation facility, if you are using a process something where you have a lot of complex excipients or APIs are being used, like methacrylic acid or any other polymers you use, it's pretty difficult to clean from the equipment surfaces. So as you keep a longer duration for cleaning, the buildup of the residues will be more. So it is purely dependent on what type of products and processes you are using in your manufacturing facility and what type of cleaning methods you are using for your routine cleaning. And based on that, some of them do every 6 months,
I
some
think
of them do every 6 months, depending upon number of batches, number of cycles. So it is purely a scientific rationale you have to make out of what exactly you are manufacturing. So there is no specific guidance. So periodical means it is, you have to define what is periodical for you. Maybe for me it could be 1 month, maybe for you it could be 2 months, maybe somebody else it could be 3 months. So periodical means not too far. 1 year cannot be called as periodical studies, or 2 year cannot be a periodical study. So periodical has its meaning of not so frequent, but it should be done frequently, maybe with a reasonable time of 1 or 2 months or 3 months or 6 months. So that could be as a periodical. But yes, provided you have to have all your scientific data as a backup to determine or justify why you have decided this is my periodical study which is required. So for example, you have multi-product facility where one group of equipment may need a 6-month study, one group of equipment may need a 3-month study, depending upon what type of product you manufacture. So that's what the take I have on this.
Thomas or Dr. Kiran, do you want to add to it?
Yeah.
Yeah.
No, I think it's absolutely right what was just outlined by Jakumar. But also what we see now happening, that if the cleaning process understanding is getting due to periodic verification, like, okay, I will do this evaluation every month now at the beginning because I do not know where I am. If I gain more data and I see after 6 months, oh, I have constantly the same results and I have a very good control of my process. So why should I continue to do this every month? Maybe I will expand it to every 2 months. And then
Yes.
after 2 years, you see, oh, I have still the same results. I have a very good equipment. I have a good maintenance in my objects. So why should I not go to extend it? So you need to get to a reason to say why you have a specific frequency and why you do not want to have it more detailed look. So that's really the aim also of the ongoing cleaning validation, that you have the assurance that you are dealing with a process and you can really get to the data that you show. Yeah, I have my process under control.
I think we can also integrate it with the risk management plan for cleaning validation
Absolutely.
that we make.
Yeah.
Absolutely. Yeah.
One,
Okay.
that's the thing which I would like to add. In fact, as both of the speakers have been mentioned very rightly. And coming to the question, if I have to rightly understand about the cleaning periodically, there are two things. One, Tom has mentioned about the periodical verification of the cleaning process. And second thing, Jacob said mentioned about the periodical cleaning well, which applies only within the campaign of the same product of the same stage. So the question who has asked this, but for them, my additional input from both the speakers, apart from the both the speakers, is first we have to very much rightly understand the process and its accumulation and its degradation, as well as through a risk assessment process, and how long it takes. And this has to be established with the analytical data, which supports to your time cycle of periodically what we have decided. Either it may be a batches, after some batches, or after some runs, or after a period of like 1 month, 2 month, in a year, whatever. So this has to be followed.
Yeah.
Uh, Thank you so much. Abhay, can we move on to the next question? Can compression machine die? Oh, this was, I think this I had sent it to,
Yes.
uh,
Yes. I did see
you as
this
earlier.
question. Yeah.
Yes, yes.
Yeah. So,
Diane punches.
yeah, because no, you cannot include in the equipment train because you are using a different cleaning method altogether here. Because
Correct.
during your routine cleaning process, if you are not using IPA and you are using a separate method for cleaning your dies and punches, you should have a separate cleaning evaluation for that process. Because the moment you changes your cleaning method, you should have a separate cleaning validation for that. So because the efficiency of cleaning changes based on the cleaning agents what you use. So it's better to have a separate study. If you are not using the same IPA for your equipment cleaning, you should have separate. If you are using the same process of cleaning compared to the dice and punches for your equipments, you can include into the same equipment train. So it is as simple as that.
Uh, Dr. Kiran, Thomas, do you
I
have
fully
anything
agree
to add?
with Jayakumar sir. The same thing like which applies if the cleaning solvent is completely different, then definitely you cannot consider it into the equipment train. If it is same, yes, for sure we have to consider that as part of equipment train, and we can. And there is one point, other thing which I also received as part of question, that surface area, how it has to be considered. It is very much easy that from where you're procuring, the manufacturer will give you the complete details of the surface area.
Yes.
So with that, you can consider.
Yeah.
Thank you. Abhay, can we move on to the next question? How direct sampling to be done in such cases for dies and punches again?
Yeah.
Surface area. I think Dr. Kiran just answered the
Yes.
question. Abhay, can we move on to the next one? Can we select vitamin formulation as worst case product? Is there any guideline reference for vitamin product cleaning validation?
This question is again a vague question. Reason behind it depends on the product matrix and the product basket, and if it is a multi-product facility. Of course, the question has come from multi-product facility, as per my understanding. But whereas vitamins comes under the food supplements, so what is the product, other products which they have, it depends upon that, and never be a vitamin product as a worst case until unless other products are very much same category.
Yeah.
Jayakumar sir, you can add
Thomas,
something on that.
yeah. Thomas has got any comments on that?
Yeah.
Are there any
No,
specific guidelines as the question was asking? Okay.
There is
yeah,
no
the food regulation. So
food
so you can
regulation.
find in the
Only the
food
food regulation.
regulation how much of supplements you can add into a foodstuff.
Correct.
This is
Correct.
what you might use as a reference. But if you look to the activity of vitamins, even at very low concentrations, they can give you a response in the body. So, and if you need to exclude that, and this is why Dr. Karan just said, this is a kind of evaluation of what other products are used. If there is a risk that these low traces of vitamins could cause any adverse effect for your next product or for patients taking your next product, then it's something what you need to consider if you will produce it in the same facility or not. Because I think these kind of things is nearly in the same discussion of what we get into when it's coming to allergens. So allergic reactions
Yeah.
can happen with a lot of different substances. And if you want to exclude that you have certain materials in the equipment, then you need to make sure that you are free of these kind of substances, because these kind of ingredients can really be active at already very low concentrations, but they do not really have a pharmacological effect. It's
Yeah.
more like that they get a response in the body, and this is why you cannot find any pharmacological regulation about it.
Exactly. Exactly. So I was about to tell that because therapeutically the pharmacological effect is not there for the vitamins. Secondly, the cleaning validation study itself is originated for the food industry, not from the pharmaceutical
Pharma.
industry. So it started from the food industry, then transformed into pharmaceutical industry. So vitamins are not considered to be a worst case, provided you are limiting it. Like Thomas told, it should not be allergic to the next levels. You have to control it. But and if you look at most of the vitamins, if you go with the current PD requirements, most of the vitamins are a very high PD values. So it doesn't fall into the category of a worst case molecule itself. So that will not be a candidate for cleaning validation.
Mhm. I think this is a very good point that you just raised. So if you look to the PDE, this is where you get an adverse effect, and
Yeah.
you do not want to get an effect of the vitamins. So, and
Right.
if you are producing the same categories, and we, for example, know that vitamin E is a little bit hard to clean because it's only fat soluble, these kind of things. But what would be a trace of this vitamin in the next
Exactly.
vitamin or mineral product be of a risk or not? This is what you need to evaluate.
Absolutely.
Abhay, can we move on to the next question? How to calculate unit dose for topical spray solution preparation? For topical cream can be considered fingertip unit FTU. Can FTU apply to topical spray solution for macro calculation?
Why you are considering still as a unit dose? Because dose criteria has been not been considered now for the macro calculations. Basically, it is all based on the PD considerations. All those traditional methods of calculating the macros are getting removed from the new for the new concept. So still I will answer how to calculate unit dose for topical spray, because when you are having any spray system, you are determining the dose per spray. Now it's up to you how much spray you are applying, because patient to patient it is going to vary. I may apply, say, for a pain reliever, I may do one spray, the second may do two sprays, the fourth may do four sprays at a time. So it's pretty difficult to determine that. So you cannot go with the dose criteria for topical sprays or creams. You have to go with, and it is a tropical product wherein the risk of cross contamination, what is the risk to the next level, because that is also a matter of concern. So dose criteria should not be a matter of discussion for tropical products. You have to go with the PDE. What is the level of contamination from one product to another can cause a uh, or an impact to the next product and to the user, that is to be calculated, not based on the dose price. So that's what my take. If Thomas has got, or Thomas or Dr. Kiran has got anything to add on that, so okay.
Mhm.
I absolutely
Yeah.
agree with you, sir. In fact, the dose unit dosage consideration at this moment, which is having conventional method of calculating or carrying out this activity, has been replaced with PD health based limits
Uh,
now at the moment.
Absolutely, yeah.
So
Mhm.
your explanation is absolutely spot on.
Yeah. Yeah, and I would also think that you need to identify what is the standard usage of the product. So you will have a leaflet telling the patient how often he should use these kind of things and so on. And I'm currently having a discussion with people from the personal care section. They also start to go into cleaning validation. And then it was the question, oh, if I produce a lipstick, how often is that applied? So and then they say, okay, ma'am, some people do it only in the morning. The others say, okay, and then I need to refresh it after breakfast, after lunch, and even in the evening. So maybe the worst case assumption is then, okay, I apply 6 times. So and this is what I will take then into account, and I will not go to the extremes that I say, okay, and I will be on the festival and I need to do it 10 times. Yeah. So these kind of things is what you really need to take into account.
Yeah.
Thank you so much. That was a very interesting point that you made, Thomas. Yeah.
Thanks.
Abhay, can we move on to the next question, please? Is it required to perform separate cleaning validation metrics for oral liquid solutions and oral suspension, wherein they are in a shared equipment?
Is it required to perform separate cleaning validation metrics for oral liquid solutions and oral suspension where the shared equipments are used? Again, what is your metric says? See, most of the product where it is in the liquid form, that means you are dissolving the entire content into water, and either that is soluble in water or any other media what you are using. And when you are making suspensions, mostly the API or the active ingredients are in a suspension form. That means you are using a media where it is in a suspended form and the API is not soluble in water. So maybe the solubility of that particular drug may not be as good as like a liquid. So definitely you should have a separate consideration for when you are doing cleaning validation for different, because these two are two different, I know, uh, formulations where the risk of carryover from a clear liquid is very less compared to a suspension, because you have suspended particles on the equipment. And if you are using the same cleaning method for both type of products, for liquids as well as for suspensions, you can consider the worst case out of your total metrics. But if you have two different cleaning methods, you should have separate cleaning validation for both the type of formulation. So that is what my take is on that.
Yeah. Yeah, and if you are going to assess the solubility, what is very often for the worst case assumptions, yeah, really I would say the precondition what Giacomo just really outlined is that you need to get to the point that you are using the same cleaning process for both of the substances. Then you are allowed to look at them together from the cleaning perspective, and then
Yeah,
it's on your worst case metrics. Do you consider pharmacological effect? You consider the cleanability maybe because it's insoluble matter. You have the solubility to take into account, or even the potency of the drug. So how low is the dose criteria and so on. And if you then have your mindset on what is the most critical one for me, and what I see very often is that people go to the pharmacological effects and so on, so that they consider that as very important, then it might not come out as a worst case product. So it's really depending on your situation. But the prerequisite is that you have the same cleaning process running for all the products when you want to group them
that's
together.
right. That's right.
My simple explanation on this particular aspect is, yes, it is obviously required. Reason behind, even for the same product, you're doing cleaning even batch to batch and as well as periodically. When you are applying for the same product, for the same farm, for the same stage, why can't it be applied for even different farm? Because even at different farm, how during the progression of time, or by the progression of some cycles, you don't know the how accumulation and contaminants being carried out. So it's obviously required.
Yeah.
Thank you so much. Abhay, can we move on to the next question? Again, we are going back to dies and punctures. Can compression
Yeah, I think
machine
I think we have already answered this.
answered this? Yes. Abhay, can we move on to next? How direct sampling to be done in such cases?
I think we have
think
already
this,
answered.
yeah, this
Yeah.
is answered.
Yeah.
Is it required to do
Clean
CEHT
equipment
and
whole time,
DHT study?
dirty equipment whole time
It's
studies.
required. It's required to do. It's required to do. Again, see, the question itself is having an answer, like clean equipment holding time and dirty equipment holding time. It again depends upon the critical equipment which is being used and non-critical equipment which is being used, and the critical process which is being carried out at the same equipment. Though the same equipment or ideal equipment you have in another place within the same process where the critical process is not being carried out in that, irrespective of that, you have to establish, determine, demonstrate that how clean your equipment is, and what is the time defined in terms of recleaning after either in dirty holding study or cleaning holding study, clean equipment holding study, that has to be established for each equipment of entire product line. And as well as you have to once demonstrate and then fix the timeline for the process or for the group of equipments or for group of the products. Based on that, you have to decide either worst case of the time considering the multi-product facility, or if it is a dedicated, again you can consider as a product wise.
Mhm. Yeah. So,
Yeah,
so for me, the clean equipment whole time is really to be evaluated on the site because it's depending on your surrounding on the manufacturing area. Is it very dusty? Do you close all the
right.
equipment? Is it something what you store in an in an area where you wrap it in plastic, for example, and it's very good dried before? So then you might consider, okay, the clean whole time is really for me something to prevent that there is a risk for the next product due to these kind of powders or any things what come from the environment, and maybe I have to look to the microbiological status of these surfaces.
Yeah.
The dirty whole time is sometimes really in discussion now because I want to give one example what we have made lately for a customer here. They wanted to establish a dirty hold time for 5 days. And they said, okay, it could be long weekend, something like that. This is why an equipment can stand for 5 days dirty and will not be processed for cleaning. We were then outlining that this is like a suspension vessel for the tablet coating. So the result after the process of manufacturing has stopped was that there was a dried on coating material in the suspension vessel. Then we were in discussion, will this dried on coating residue change over the time, over the 5 days or not? So we were doing some studies on, yeah, have the dried on coupons for cleaning studies after 1 day, after 2 days, after 5 days. Is there really a difference in cleaning? And we could identify, no, it's not the case. This situation, were really in discussion now with this customer that they say we need this equipment for production, and we do not want to hold all the suspension vessels. We want to validate for 5 days in the equipment. So this is why here we said, okay, in this case there is a possibility maybe to shorten down the studies in the site to say, oh, it's a completely dried on residue. We do not have very high microbiological risks, something like that. So we want to go for 1 or 2 days of period of dirty hold time, and it will be representing also 5 days because this is what we saw in practice in the laboratory cleaning studies. It would be a complete different situation what we have had a discussion with a biotech company when they make some fermentation broth or something like that. After 5 days, everything is growing heavily in there. So there you get a completely different residue and you have a different kind of cleaning afterwards because yeah, the sugar and these kind of nutritions are eaten up by microorganisms. So it can really change. So these would be the things of what you need to consider. And I would always be a fan of, like Dr. Kiran said, to make the also dirty whole time studies in the time frame that you want to evaluate, because then you have the real data. But if you really have high pressure on production needs and so on, and you want to see where is the possibility of maybe having the equipment available earlier, then you should need to go for studies to say, oh, is there a change of the residue on the equipment over the hold time study or not?
Yeah, I think
While
that's, I don't have any comments further on that. Yeah.
I think while we are on the DEHT study, there's one more question which is talking about what shall be the acceptance criteria for degradation of products and impurity profile.
Oh, also wonderful question. Uh, if you have any separate specification for degradation, then that's because other than the product, the interest will be the product purity and the impurities of the defined, specified, and unspecified. See, this is the interest of estimation of dirty holding study where the degradation takes place. If your every study gives you some picture that in some environmental conditions, either with foods degradation of oxidizing, or even with water, whatever peroxide, in any conditions, heat and temperature, uh, with any kind of extreme conditions, if your producting is getting degraded, the same simulation, either you are identifying with the manufacturing equipment, which may lead to contamination or unwanted impurities to be in a elevated level or more than the expected level, That is the interest of estimation. So there is no other specification other than the product specification for this degradation.
That's right. That's right. And also, perhaps you may have to look at if you have a probable, impurities, and if you know the toxicological level of those impurities, know about that. And if you find some of your impurities are highly toxic, maybe you may have to think in that front as well. So if they are, you have the toxicological data mostly available during your post degradation studies, and when you have to identify all those impurities after having 0.05 percentage threshold, then you have to go for identification. You have to do all those toxicological studies and all. So based on that, you can determine if you want even a stringent limit compared to your product specification. So nothing beyond the product specification. Even you need to think beyond, even below the product specification. So that should be the level of criteria you should keep it.
There is one more follow-up question to this by Mr. Vikas Gaikwad. He is saying, is it okay? Not this one. Away. There is one more where he's talking about, is it DEHT and CEHT campaign study visual inspection qualification? Yes. Need to be done for all products or only for the worst case product?
I'm not clear about the question. Is it DHT, CHT campaign study visual inspection qualification need to be done for all the products?
Not necessarily, not required, because you know your product. Say, for example, if it is in a formulation facility, like an OSD, because in a liquid facility, it is pretty easy process of cleaning. Let us consider the worst case scenario, like in an OSD facility, if you have a product which is based on a direct blending process where you are adding all the components into a blender, blending it, removing it, compressing it, and packaging it, where you use a granulation process, a wet granulation process, whether you use a solvent or water as a granulating agent, and you have your binding agents which is sticking onto the equipment surface. So it is pretty difficult to remove from the equipment surface. So you should have a worst case metrics prepared for your dirty hold time studies, dirty equipment hold time studies. And based on that, you should study it, not necessarily for all the products. So you have to decide, you know your product better. So when you have a product development happening, your R&D should give all those information to your facility. What should be the cleaning method is to be used for this product, and which is very difficult to, clean, because I am happy to share with the forum that we have worked with Thomas and team, with the Ecolab team, in developing cleaning methods for different products, for different cleaning methods depending upon the type of product what we manufacture. Where we use polymers, so we have to use a different method, where the dirty hold time study plays a vital role. So because it's sticking to the equipment surface, and as long as it is sticking to the equipment surface, it there is a potential probability that the degradants can happen on the equipment surface over a period of time, because it is in a wet condition which can accelerate the degradation of the product. Then it is very important for you to study such products compared to a direct blending process. Similarly is the case with an API. So whether the API is highly soluble, and whether the API is not soluble in water, or it is soluble in some other solvents. So depending upon the nature of product, you need to decide something like you are making a cleaning validation matrix for the worst case selection. Something similar you have to work for your dirty hold time studies as well. Not necessarily for all the product.
And further add on to this point, yes, in some cases, like especially if any of the coloring agents or coloring products are being carried out in the same facility, it depends. Like visual inspection has to be carried out, unlike to non-color and color products. But
Yeah,
however, all this study definitely depends upon a scientific rational and risk assessment.
absolutely.
Thank you so much. Abhay, can we move on to the next question, please? Because I hope that was answered. If there is any follow-up question, please again put in the Q&A tab and we can revisit the question. Until then, moving on to
This
the
is a
next question.
this is a
Yeah,
common question where industry is having. Is it required to perform the
yeah,
cleaning validation on a product which are having less than 10 micron instance 10 ppm? It seems the question PDE or worst case least can be considered. Of course, it's least is the considered as a worst case in any scenario, provided your analytical method should capable to detect LOD and LOQ lower than the particular limit. Otherwise, your LOD and LOQ values are more than your limit, it is not justified. Many of the citations have been issued for the same. And considering the worst case of 10 ppm, uh, which is practically possible, which sometimes practically not possible, that we have to reevaluate our cleaning process. That is the reason many of the current guidelines revised for looking of our cleaning process review, re-review with respect to the analytical perspective, and also as part of assessment, risk assessment, which we need to do based on the current requirements.
yeah. So to add upon that, any molecule which is having PD less than 10 microgram considered to be a risk item. That means when you manufacture that item, because it's highly, potential for the effect on the next product, and the patient is, highly uh, possible if there is a carryover for more than the allowable limit to the next product. So the requirement is to determine whether it requires a cleaning validation or whether you wanted to do a cleaning verification. But it is generally expected that if any value which is less than 10 microgram needs to be verified for its cleaning effectiveness, though you have a worst case, say for example, you have a worst case molecule of 2 microgram at your site and you did a complete cleaning validation study, including 3 complete cycles with operator variability, all those things you have done it. But if you have a candidate 5 pp 5 microgram or 6 microgram, it is a general expectation that it has to be studied for its effectiveness of cleaning, to make sure that your existing cleaning methods are able to remove any residue of that from the equipment surface. So no guidance other than MHRA is insisting on that as of now, but MHRA is expecting that anything below 10 microgram needs to be verified for its effectiveness of cleaning. So that is the general expectation now. So if it is in my case, yes, in our case, we do it. Anything below 10 microgram, we do it.
Thank you so much. Abhay, can we move on to the next question? Should VRL be established for all the products?
I think we have answered in a different way for the different question earlier.
With the visual residue limit, yeah.
Yeah. Okay. Is any study available to determine worst case location of equipment?
There
Any
will be no
guideline
study
or
available
reference?
in general, but you have to study at your facility based on your equipment train and design, and as well as the product accumulation and worst case of worst area of cleaning, hard to reach areas and critical to reach areas or critical to clean areas. So many things you have to consider. The best approach for this study is risk assessment. You have to carry out for your own products at your own facility.
Yeah. Yeah, Dr. Kiran is absolutely right, and I think what would be, just to add one comment to that, what I always see that people are tending to say, "Oh, I passed the riboflavin test and all is good," or they even consider, "Oh, I have maybe a spray shadow here on the top of the reactor, and this is my worst case area." You need also to consider if even product is reaching there, because if you have, like in the worst case situation, your dome of the mixer even not dirty, let's say, there is no residue. Why should this be a worst case step? And I very often see that people forget in the risk assessment the process parameters. If you, for example, heat up with a jacket on the mixer, the product, and you have an introduction of a powder, and then powder hits the hot surface, so it dries on heavily. And this kind of situations is where we would say, maybe this could be a worst case identification, because from the cleaning perspective, this residue now dried on at hot temperatures is much harder to remove than a spray shadow on top of the mixer, because this is the kind of risk assessment what you need to do here. This evaluation needs to be done, and it is always, from my perspective, a very good advice to ask the operators, where do they see issues in cleaning, or where do they see high amounts of product residues still in the equipment, for a consideration of making a worst case spot. And if we even go to the digital world, you can even mimic some geometries and so on
Yes.
to see where you might have a spray shadow, and these kind of things can be done, but do not forget your process parameters.
Yeah, it also plays a very important role in designing your equipments. When you have a design space, you should really look at whether my equipment design is such a way that it is very easily cleanable. Because most of the time, what happens at the design space, we don't pay more focus to the design of the equipment, and once it reaches to the site and when we start using it, we determine that no, these equipments are not easily cleanable. Then you have to read. So it's very important to include your engineer, your project team, and your quality team, and the cleaning validation group. And the validation group has to sit together. Whenever you get a design from an equipment vendor, you should really see whether it is easily cleanable. So that is a first step of. That's why you should have a master contamination and cross contamination strategy in place, where you should design all those, or you should determine all those factors. Then you should go ahead. So that will make your life very easy than doing it when you installed everything and now you are saying it is not cleanable. Then it's too late for you to address.
And this is a very important point. So what you just said, Giacomo, if you have a system in place and now somebody is coming to you and saying, oh, I have here a paste you can apply to identify if you have a spray shadow or not, please consider, what will you do if there is an identification of a worst case spot? So will you need to recall all your batches before because you have not considered this for validation? Or is it something where you can say, I have the data that I was good before, that is maybe only an additional point for me to consider. So it's very good to do this when you install an equipment and not to do it after like 5 years of operation.
Absolutely. Absolutely.
Thank you. Abhay, can we move on to the next question? What is the frequency to inspect pipeline?
During the product changeovers, it's basically for sure. Sir, you can add.
Can I ask Reshma
So
to actually?
can you slightly elaborate?
Yeah.
Yeah,
Reshma,
I
can you
mean,
please
which type of
elaborate the
pipeline
question?
they're talking about?
Till then, can we move on to the next question and we'll wait for Reshma's comment to come.
Yeah.
Abhay?
If I correctly understand that about the pipelines, I think those are the product transfer pipelines.
If
Vishali, go
Okay.
ahead.
So
Yeah.
who want to take this?
3,000 liters vessel.
Yeah.
This is a
So how do you
this is
want
a
to,
very good
who wanted to
question.
take this?
No,
Yeah.
I would say you really need to do a kind of inspection, and if you have a 3,000-liter vessel, it needs to be verified what you want to expect during the validation. During the validation period, you might use a mirror to look behind the stirrer or from the bottom side and so on. This is what you will do for the validation, but for the ongoing verification that everything is clean, you need to identify how to do it. So to what points or to what spots the operators should have a look at. Maybe they use a torch or light or whatever to make more visibility of residues. But this kind of evaluation of what spots to look at, this needs to be done in with the cleaning validation protocol and needs to be intensively done, and also the inspection maybe of the circulation line with the boroscope or whatever when you do the validation. And you need to consider then in my daily activities, I'm controlling with the rinse sample or with an online measurement of TOC, conductivity, whatever, that I have the process still under control. And I will review these critical areas, what I cannot reach maybe in the normal circumstances, if I do a verification of my validated process.
Yeah. So in addition to that, nowadays there are a lot of advanced equipments available, because if you cannot see something with your naked eye, there are magnifiers available, electronic magnifiers available, where you can reach up to that area and you can see it an enlarged view of that portion of an equipment if it is directly visible to you. Otherwise, there are a lot of systems like people are using mirrors. You can install mirrors on a stick and then you can look at the mirror. If you cannot see it directly with your naked eye, you can take angles on a mirror and then you can focus on that area and see how it happens. So there are different methodologies for visualizing a high size equipment. So depending upon the design, you can use different type of tools. You can use, it is something like you are using a binocular to see something at far. No, you wanted to see it closer to you. It is something like that. So you can use different piece of equipments. And nowadays there are a lot of equipments available to use for such type of purposes. So you can opt anyone which is suits to your equipment and facility.
And
Can AR, VR or sort of technologies help in this as
Yes,
well?
yes,
Okay.
I agree, but one more add-on to this question is very much vague question. Reason behind 3,000 liters vessel, it may be of any shape. First
yeah,
thing.
that's
So,
what.
because in the formulation it will be an open-ended, and
Correct.
like see, for example, if I have to say FBD ball, it may contains again the size of the ball which is very much visible to naked
Yeah,
eye at any given point. But whereas coming to the process equipments, coming to the bioprocessing or APIs like reactors, and some of the more than, if I have to say about my past experience, that I have been gone through 32 KL reactor as well. As Jayakumar said very clearly, there are equipments available at this moment, but again, this has to be covered with your risk assessment process. How you are going to declare that your equipment is contaminant free, on what basis
yeah,
and what kind of measures and what kind of authentication you're giving, and from where you're picking the samples exactly, on what rationale. That again depends upon the design of the equipment, and again the experience on that particular equipment. So it is more subjective and case specific. But however, if I have to say about, there are some prerequisites which you have to ensure to witness whether this accumulation is possible, I mean, there or not, and it is residual free or not. And after that, analytical evaluation and everything will give you based on the sample you picked up.
yeah, yeah.
Thank you so much. We are actually towards the end of the time, but we have few more questions left. Can we take a couple of more
Yeah, yeah,
questions with
sure.
all your permission?
Um,
Okay.
yeah.
Abhay, can we move on to the next question? As per recent US FDA guidance, one of the root cause was presence of nitrosamine impurities in fresh solvent, but it is not specified which impurities were detected. If we
It's
need
a
to
very good
test the
question.
fresh solvent,
It is a very good
I think
question.
Dr.
But
Kiran, you should
again, but
definitely
again,
answer.
I'm saying this is the fresh solvent from where you have procured, and what kind of authentication or certification you have received for that. That again depends upon your sourcing and internal thing. But however, if I have to see about the risk assessment of nitrosamines, your all inputs has to be subjected. And coming to the process criticality with respect to the nitrosamines, the prone of nitrosamine impurities with respect to your inputs, you have to first initially verify, estimate, evaluate, and then not only theoretically, if there is a prone of such kind of this thing, then you have to understand your process criticality. And coming to the more subjective one, what kind of nitrosamine has to be tested? It again depends upon the fresh solvent which you have carried out. Initially, you have not found anything where there is a requirement of nitrosamine impurities evaluation as the fresh solvent, and why it has been subjected, and how we have arrived that from the fresh solvent this nitrosamine has been attained. If it has been attained, what nitrosamine you have identified, it depends upon that what to be looked into.
Yeah. Also, it depends upon the process what the manufacturer has used to manufacture the fresh solvent, because it can come from the starting material what they have used for making the fresh solvent, maybe the equipment what they have used for the fresh solvent, some leachables which got into the solvent during the processing, and you have to look at the structure, the chemical reaction behind, or the structure of that particular solvent. So by doing that all studies, you will get mostly a probable type of impurities which is possible in that particular product. So your aim is to focus on that first. Then whatever methodology you use, whether you use an LCMS or a GCMS for your identification of those impurities, you can fairly get an idea it matches with what type of mass of that particular impurity. So then you can reach down to the probable impurities in your fresh solvent, and then you can determine what should be tested for that. So it all depends upon the route of synthesis by the manufacturer to manufacture that fresh solvent, the equipment they have used, and what are the structure of those molecules, and what is the probability of getting found the nitrosamine impurity at that fresh solvent. So those factors needs to be studied to determine, because what is happening now with the APIs is exactly the same process is being done. What is the structure of the molecule? What can impact, whether temperature, humidity, the structure, free amines, free nitrates available is causing the formation of the impurity. So everything is studied, then we are reaching out to these are the probable impurities for this API. Then I need to monitor these impurities. So the same process has to be done for the fresh solvents as well.
Thank you. Abhay, can we move on to the next question? Just before we go to the next question, there are a few questions, I think, in the chat section as well, Abhay, if you can just see that. Moving on, whether cleaning validation is required for product non-contact part of FBP duct. If yes, how to calculate acceptance criteria?
If duct is part of your equipment, see, question is if the duct, it again subjective. If equipment contains, design contains the whole thing, and that comes under the part of equipment which you can disassemble, or for each and every batch during the product changeovers or so, you have to consider not as part of product requirement and product specification in routine, but however as part of contamination control strategy on a random basis, you can evaluate for information not to have more accumulation through that product non-contact part, as well as even non-routine equipment parts which you are touching.
Yeah, Thomas, you have anything?
Yeah, the European Medicine Agency has in their question and answer session for the health-based exposure limits also a kind of outline that they say if there is a possibility that from the outside, even if it's not a product contact surface, but that something get detached and can get into your product, then you need to include this kind of surfaces in the visual clean inspection. So it should be at least visual clean. So if you are above a reactor or a mixer or whatever, and you have like dust and on the top of the ceiling, then there is a potential that this falls into it, and you need to clean it up. So this is the kind of evaluation, and then you even do not need to make a limit calculation for that, but you need to make sure that it's visually clean.
Yeah.
And add on to that, previous discussion on the point of equipment cleaning, where both the speakers have quoted about many of the techniques and equipment, sophisticated equipments have been come up with in the market now. One is telescopic, telescopic camera, robotic camera, which goes to the depth, any depth, and can identify any kind of powder accumulation or so. These kind of things usually on a not on routine basis, at least on a long campaign, lengthy products cleaning time, product changeover cleaning time. Many of the industries are adapting, companies are adapting to avoid such kind of unwanted contamination into the during the inspections, not to have any citations. So these are contamination control strategies implementing.
Yeah.
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 Abhay, can we move on to the next question? I think we have
I think we
answered
have
this
answered
already.
this.
Yeah. Yeah.
Mhm.
If an anomaly is found on equipment and that is within limit, do we need to perform a cleaning verification?
What anomaly on equipment? I don't understand the question.
Yeah. The question is not so clear to answer this.
Mhm.
Reshma, can you?
I would think that what they call anomaly is if it's within the limits. So if I think of a cleaning process, for example, and you do not reach a certain temperature, you identify you need to get at 80 degrees and maybe it's plus minus 5 degrees C, and you make a cleaning then at 78 degrees, you are within the limits, but you are not, you are not having the 80 degrees, what is the normal process. So then my answer would be you need to make sure that you set the limits into the way that you always have the best result. So if you say you can reach 80 plus minus 5 degrees, there must be a reason to do that. So and with if you're within these limits, you need to be acceptable. So that means the validation would be then done on the worst case. So you would do the validation at 75 degrees and you achieve for 80 degrees for cleaning, and then this anomaly is within the limits and would be acceptable because you validate the process in a different way. If you not do this, then you might need to do a risk assessment. What has happened, and is this something what you can say it's okay, or you need to really verify that the cleaning was successful? From my perspective.
Okay.
Uh, thank you. Abhay, is there any other question? If any, in case of excipient to be considered in cleaning validation. If any case
In any case, excipient to be considered in
considered
cleaning.
in cleaning validation.
It all depends upon what type of excipient you are looking for. Like, there are few excipients which is very difficult to clean from the equipment surface. Okay, like I quoted an example of polymers. They are very difficult to clean from the equipment surface. Maybe your API might have removed from your equipment surface, but your excipient residues may still remain on the equipment surface. And once your equipment cleaning process is done and once the equipment is dry, you can see white spots or white patches on your equipment surface. That means your cleaning methods were not able to completely clean your excipient residues from the equipment surface. So yes, you should also consider when you are talking about cleaning validation, it is not only the API what we are aiming at, so it should be visually clean. So maybe excipients may not have a therapeutic impact on the carryover to the next product, but visually clean is a must for any equipment cleaning process. So you have to consider as a whole the entire metrics into the cleaning validation, not only that your API, maybe your cleaning validation samples might have got zero ppm or 0 microgram for your API, but still you see your equipment is not visually clean because of the excipient residues still on the equipment surface. That cleaning validation is not effective. So you have to consider your equipment residues as well removed from your equipment surface.
Yeah. And
Yes.
it's really funny that this question was raised, because last week I got a question from one of our customers in Switzerland. He had an inspection, and the inspector was asking, "Oh, you are using magnesium stearate. This is insoluble. How you make sure that during the cleaning you can clean off this insoluble material?" And so that was the question from him. And I said, yeah, but you are checking exactly visual clean. So you would see magnesium stearate on the surface. And
Exactly.
at the end of the day, we have made some studies based on some raw materials, and we could convince that this kind of cleaning action with surfactants is emulgating these insoluble matters and bringing out of the system the raw material, this excipient, yeah. And but at the end of the day, Giacomo, I would completely follow your logic and say, yeah, this white powder is insoluble, we know, but if we don't see it on the surface, it's gone. It's yeah.
Yeah.
And answer to this question in a different way, that even it is an excipient, it's a contaminant to it will be a contaminant to other product. So definitely
Yeah.
it has to be considered. And I
Yeah.
have come across to other one of the typical case, like in API, majority of stages, some of the stages in the process will be as in situ stages, whereby
Yeah.
cleaning process will not be exactly targeted, as the working standard will not be available. And in those cases, what we have to do is, we may not go to the specific testing methods. You can go for non-specific testing methods as well. Like one of the cases, resin we are carrying out, that resin is very much visibly it will be clean, it appears to be visibly very clean, but you defining that, whether it is clean or judging it is very difficult. So I have introduced some of the non-specific method, like conductivity is one of the method, and
Yeah.
pH is another. So these kind of methods you can introduce. We have to explore our options, not only for the specific and also for the non-specific, so that we will get solution.
Absolutely.
Correct. Yeah.
Absolutely. Thank you. Thank you so much. I think we have answered all the questions that have come up now. If there are any further questions, please post them on the cleaning validation group that we have, and we can definitely reach out to our experts and get the questions answered. And thank you so much, Jayakumar. Thank you so much, Thomas, Dr. Kiran, for spending this time with us
Thank you.
and
Thank you.
sharing your experiences with us.
Thank you. Thank you. Bye-bye.
Thank you.
Pleasure.
Thank you
I
so
hope
much.
it was of help.
Thank you.
Thanks
Bye-bye.
very much.
Thank
Bye
you.
bye. See you.